Good morning, y'all. Good morning. All right. Hopefully, a few more people come in here. Yeah, it is what it is because it's summer. All right. So we need to push on. We have one lecture and digestive system left to get through, and I think you guys are ready for it. Honestly, you've been doing a fantastic job keeping up. Not just keeping up. I hope you guys notice there's a method to my madness, right? It's like at first I'm introducing pieces of the puzzle, and then I build on it and I build on it, and the next thing you guys know, you're answering all these questions. It's crazy. No, there definitely is. There definitely is. Especially if you're able to how to it gets a little bit harder if you're coming in and out of class, right, Melissa? But if you're here listening to the questions that I'm asking, they all are interacting. And I know you listen to every single lecture, so that helps too. Okay, so method to my madness. Let's remember, what are the 6 processes? What are the 6 processes of the digestive system? Let's throw them out there. You guys type them out, all 6. I'll give you 30 seconds. Let's go ahead and try, Andy, go ahead and try to type all 6 in chat in one line. Do your best. If you get 4 out of 6, great. Try to get 6 out of 6, though. And don't press enter yet. Don't press enter yet. All right, go ahead and press enter. Good, great, awesome job. Perfect. Love it. Yeah, even if you only got a few of them, that's great because start, right? And then you could work on getting all 6 down next time. But this is great. Yeah, the first thing I would write is ingestion, but it doesn't matter what order you put these things in. Ingestion, digestion, both chemical and mechanical. Motility, because we got to move things through the system. Secretion, because we're constantly pumping liquid, basically solutions into the system. We're talking about saliva to just straight bicarb-rich mucus to hydrochloric acid to different enzymes and digestive juices and pancreatic juices and bile. Lots of things are getting secreted into the system. What's being secreted into the system is helping with digestion, either by lubricating the system or by being the enzymes that actually do the digestion. Once we digest the food into small enough pieces, and we're going to be talking about that a lot today, once we digest the food into small enough pieces, and I'm talking molecule-wise pieces, right? They have to be the monomers. They got to be the smallest base unit for that particular type of biomolecule. Now we can absorb them. Transporters can't just absorb big old things. So your body's literally taking proteins down to single amino acid levels. Your body's literally taking carbohydrates down to single monosaccharide levels. Is this making sense? We have to get them down that far before we can absorb them. Why? Secretion and absorption. If I talk about secretion, if I talk about absorption, what must we be thinking about alongside of those 2 words? We got to be thinking about these protein transporters. Protein transporters can only transport things that are small enough. End of the road, defecation, elimination, same thing. Okay, defecation, elimination, same thing. Now, we've already talked quite a bit about going through the flow, right? Literally starting at the mouth, and we've made it all the way down to the anus. We've talked about the ingestion of food. We've talked about the chemical and mechanical breakdown of food inside of the mouth. Please make sure you guys can do this. Flow it out. Secretion of saliva into the oral cavity. Swallowing all of this bolus of food is going to involve motility, peristalsis. It's going to have to go through a couple of sphincters before it can make it down into the stomach, and the stomach is making hydrochloric acid, but it's not making just hydrochloric acid, right? The presence of food in the stomach or vagal stimulation. Now we're getting G cells that are secreting gastrin. Gastrin is stimulating parietal cells to secrete hydrochloric acid. Hydrochloric acid now causes pepsinogen to be released. The hydrochloric acid's denaturing the proteins in your stomach and cleaving the ogen off of pepsinogen. It's all a big gambit, right? A lot of crazy stuff going on inside of that stomach. That flow chart is called what again? I actually never really come up with the name of it. I mean, it's hydrochloric acid secretion, stimulation. Yeah, gastric phase, that works. I like that. It's all part of the gastric phase. It's not the only thing involved in the gastric phase by any means, but it's definitely the flow chart that is a very helpful flow chart in terms of that hydrochloric acid. So let me ask you guys a question based on what I've talked about so far. What is the function of hydrochloric acid in carbohydrate digestion? Increase, decrease, no change. Let's just go there. Did I say I did say hydrochloric acid, right? So you guys got to stop overthinking. Does hydrochloric acid have anything at all to do with carbohydrate digestion? Yes or no? Is there any physiological relevance whatsoever between the two? The answer is no. Amylases digest carbohydrates. So if I ask a test question on hydrochloric acid and I throw in carbohydrates, that's a trick question, y'all, and you got to be able to say, wait a minute, there's no physiological relevance. None whatsoever. Therefore, the answer is going to be no change. If there's a physiological relevance, that's when it's an increase or decrease. Does that make sense? It's yeah, it is what it is when it comes to trick questions. They're not, I mean, they're not really trick questions. We got to be able to think about this with the patient that might be having salivary secretion issues. What is the problem? If they don't have saliva, one, swallowing is going to be pretty miserable because they're not lubricating their food. And two, we're not initializing the digestion of carbohydrates. But what you're going to find out is that's not the end of the road, but it will end up impacting total carb digestion and absorption. But all right, stomach, we got to go through that pyloric sphincter. The pyloric sphincter I like to test on. It's a pretty important sphincter. It allows the stomach to be a storage container. It's going to release little boluses of chyme into the duodenum, and then the duodenum does the large majority of the work. Duodenum and jejunum, right? Those are the ones with the largest surface areas. Those are the ones that do most of the digestion. That's occurring in the duodenum. Most of the absorption's occurring at the end of the duodenum and then through the beginning portion of the jejunum. By the time it makes it to the ileum, there's pretty much nothing left to absorb. It's almost all done. We're going to talk a lot about this portion of the tract today. All right. But gets into that cecum, some bacterial fun, through the ascending, transverse, descending, sigmoidal. This whole portion, each one of these haustrum, they are reabsorbing ions. Let's not forget that. That order of events is important. They are reabsorbing ions. They're using transporters to move ions back into the interstitial fluid and then into the hepatic portal system. And where does water want to be? With those ions. It's going to be moving with that party. And therefore, when we reabsorb ions, water goes with it. Make sense? And that is what's going to concentrate your waste. It's the reabsorption of the ions. We don't have active transporters for reabsorbing water. It doesn't work like that. Water works via osmosis. Water goes where the party's at. It doesn't have a choice. Get it, got it good? And we talked about the defecation reflex. There's both an internal and external anal sphincter. Correct. All, yeah. And then we were able to talk about that a little bit yesterday in class too. But all laxatives, that's what they're all about. They're all about trying to maintain ion concentration inside of the large intestines. So I'm not sure what Miralax exactly is. I could look it up real quick, whether it's a bolus of high concentration osmolarity. A lot of these are just big, hypertonic solutions that you can't absorb. All those milks of magnesias and stuff like that. I'm not seeing exactly what it is right off the bat. Polyethylene glycol, maybe. I don't know. Yeah, you guys can look that up too. But yeah, I don't know whether it's blocking ion absorption, which is always a possibility, or whether it's just increasing the hypertonicity of the of your chyme. All right, let's get moving though, because at this point, what we want to start to do is we want to start to tie in the rest of this idea of the liver making bile, sending it to the gallbladder. The gallbladder stores it, releases it when we need it. We're going to talk about the signals that trigger the gallbladder to do what it do. Pancreas secretes pancreatic juices. These are all of those peptidases, amylases, lipases, and nucleases, and bicarbs that all get pumped into the duodenum. Very important, all of those, including the bicarbonates, because we need to neutralize that acid. And we're going to be talking about what's happening over here in the duodenum. There are other enzymes that are going to be triggered for secretion here in what's called the brushy border or brush border of your duodenum. All right, and all of this is going to be controlled by both intrinsic and extrinsic controls. Those are words we should be pretty familiar with at this point, right? The body is controlled with a local and with hormones and nervous system. So that's what we're going to be talking about. Okay, so when we're dealing with local, what we're actually dealing with as a local is at the, I already mentioned this, but your digestive system has its own nervous system. It's its own distinct, separate nervous system called the enteric nervous system. And so within your GI tract, there is a big branching nerve plexus that's all part of this enteric nervous system. And if we trigger that and just those local reflexes, that's considered a local response, an intrinsic response. Does that make sense? If we send that information back to the brain and the brain creates a reflex, that is going to end up being extrinsic because it's gone back to the brain. If we send a hormone into the bloodstream, then that's extrinsic as well. So if it's got to go away from the GI to help regulate digestion, then it's extrinsic. Cool, we got that. Intrinsic versus extrinsic here. Nice. And what do we know about all stimuli in the human body? If you have a stimulus and your body's going to respond to that stimulus, we better have receptors for that stimulus. Make sense? Based on everything you guys have learned so far, all stimuli must have receptors or we don't respond to it. Pretty simple. So what types of receptors are found in the GI? Baroreceptors, chemoreceptors, osmoreceptors. Easy peasy lemon squeezy. We need to make sure that we respond to stretch, especially in places like the stomach. Chemoreceptors. Remember, even water is a chemical. ATP is a chemical. So things like a peptide, chemicals we can respond to. Fats, chemicals we can respond to. True story? Carbohydrates, chemicals we can respond to. So if I have a chemoreceptor, a chemical receptor who's designed to monitor for carbohydrates, boom, carbohydrates are going to trigger that chemoreceptor and send signals around, talk, 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 talk. The nervous control words to use, short reflexes versus long reflexes. Short are intrinsic. They are all within that enteric nervous system. Long, going back to the central nervous system, sending that signal back to the GI, that's going to be extrinsic. So for instance, if you smell something delicious and then your stomach starts to churn and growl and gurgle, we all know what's going on there, right? That is going from the cephalic phase and talking to the gastric and intestinal phases. So that's going to be a long reflex that's going to be extrinsic. Last but definitely not least, we're going to be learning quite a few different hormones today. This is one way to look at it, and this is another way to look at it. I actually really like this flow chart. It seems complex at first, but it's not because it's color coordinated. Green, extrinsic autonomic nervous system. So we're talking primarily the parasympathetic for activation and the sympathetic for inhibition. That should make sense, right? Rest and digest, if we're talking about excitation, we're talking about the parasympathetic nervous system because it's rest and digest. Yes, no, maybe so? And then the sympathetic nervous system is designed to shut things down, shut down blood supply, shut down smooth muscle contraction, so forth, so on, shut down hormonal release. It's really this guy right here. You can see the arrow goes to the enteric nervous system, the intrinsic nerve plexus. It goes over to the GI hormones because this autonomic nervous system helps control everything. Everything that's coming from the cephalic phase, all of these external influences, they're going to be acting via the autonomic nervous system. End of story. This flowchart is missing a couple things, but I'm not going to test you guys on those things. But I think it's important to note that gastrointestinal hormones are not just activated by receptors in the digestive tract. It's a lot more complex than that. So if you can just listen for a second, don't write anything down. GI hormones, these guys right here, they go everywhere. They're hormones going into the bloodstream, right? So they're not just acting on our local GI tract. These hormones act on fat. These hormones act on the brain. And then if you look at fat, the fat's talking back to the GI hormones. The fat's talking back to the stomach. The fat's talking back to your small intestines. And the fat's talking to your brain. Your brain is talking to the GI hormones. Your brain's talking to the stomach, and your brain's talking to the small intestines. And guess what? Your brain's talking to the fat. There is a big, huge, convoluted flow chart when it comes to the digestive system. It is not a simple system. It is not straight line flow charts. Does that make sense? It should make sense because we all eat. Eating is not simple. Your body is constantly trying to communicate. Ultimately, the central nervous system can override it all because limbic system. Damn you, limbic system. Right? Whether it's anxiety, stress, or I just want that chocolate tort. You know, tart? There we go. Is there a tort out there? I don't even know anymore. Yes, there is. Okay. I thought it sounded right. Cool. So that's kind of what this flowchart is designed for. But ultimately, what I want you guys to see is that the intrinsic enteric nervous system, the little brain, the autonomic nervous system, GI hormones, they are all going to be acting on effectors. And what are those effectors? Smooth muscles, exocrine gland cells. These are the ones that are going to be secreting pancreatic juices and bicarb and bile, all that sort of stuff. And then endocrine gland cells. So we get even more hormones like insulin, glucagon, and a big old list of other hormones that you guys need to know. Good news, it's in a table form, so if you got, your test on Sunday, you don't have to have these all memorized by then. You got a table you can look through. But we're going to work on learning each one of these today. The primary hormone that's released from the stomach is gastrin. Boom, ba boom. You guys already are familiar with gastrin, right? Which cell secretes gastrin? Awesome. Easy peasy lemon squeezy. It's the G cells that secrete gastrin. Those are stimulated by what? We went through a flowchart on this. Well, cool. I got a table for it too. What's the stimulus for the G cell activation and the secretion of gastrin into the blood? Let's get some vagal stimulation, neural reflexes, and then peptides, amino acids in that stomach. If you get food in that stomach, especially if it's a protein, we're going to start to stimulate gastric acid secretion. What's the blanket inhibitor for the entire stomach hydrochloric acid secretion? What hormone inhibits everything? Yeah, good, that's the cell. And then the hormone that does that, somatostatin. So it's all right here in this table too. So this is a nice little quick reference. Sound good? The other hormone that I will definitely find a way to test you on, it's one of my favorite digestive system hormones, is cholecystokinin. Say that 5 times fast, right? Cholecystokinin. Well, the good news is you can just abbreviate it CCK. CCK is crazy important when it comes to digestion, absorption, secretion. So it's basically, ultimately when it comes down to it, it's probably more important than gastrin. I don't know. It's hard to put these guys in a list of importance, but CCK is really, it has a lot of functions, a lot more than are listed here. In fact, let's go ahead and start writing down some of their functions using a flowchart down here on my whiteboard. But I want you guys to first look at the stimulus here. The stimulus for CCK release is going to be the presence of fatty acids and amino acids. There are 20 amino acids in the human, essentially amino acids. Some of those are triggering CCK, but typically if you eat a meal, you're going to get some of those amino acids. So what do you guys think don't look at the table, don't look. If the stimulus is the presence of fat and protein, what do you think a large role of CCK is going to be? If the stimulus is fat, if the stimulus is protein, CCK is going to help with not breaking them down, but I love that you guys went there. That's fine. It's going to help with that process, isn't it? It's going to figure out a way to communicate through the body that, hey, we got fats here. Boop, 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 boop. We got fats. We got some more proteins down here. You guys got to figure out what you're doing, right? Okay, pancreas, you do your thing. The liver, you do your brain. You got to do your fat. You do you. So that's what CCK does. It's basically this gigantic call system. It's a trigger system. So if we want to start writing out a flow chart for this, we're going to say, you eat some fat. You eat some protein. I'm just going to categorize it, broad categorize it as proteins, because it's not like I'm telling you to learn the different amino acids that trigger CCK. That's fine. All right, so we're consuming this stuff, and it's gone into the system. We start to digest those fats kinda in the mouth, not really, because we're secreting lipases in the mouth, and then it makes it down to the stomach, and those lipases are activated, and we start to break those fats down in the stomach. The proteins, they have done their thing in their stomach too, right? Because the hydrochloric acid has denatured these proteins, and the pepsin has broken them down into oligopeptides. So now let's get these fats and proteins into the duodenum, because that's the important part here, duodenum. We're getting these fats and proteins already partially digested into the duodenum, but we're not done digesting them yet. We got to do more. Well, we have chemoreceptors for these fats and for these proteins that's going to trigger the release of CCK. Okay, it's going to activate CCK. And CCK is an endocrine hormone, so it's going to go into the bloodstream. Does that make sense? Endocrine, blood, should be pretty simple at this point. So once it goes into the bloodstream, it goes a whole bunch of places. It goes a slew of places. It goes to the hypothalamus. Good old hypo, right? Hypothalamus, one of the key players in hunger. The more fats and the more proteins you eat, the more CCK is released, the more it tells your hypothalamus that you're good. I'm good. It's called satiation. If you're satiated. Satiation. So it should trigger the loss of hunger. Okay? When you're satiated, you're not hungry anymore. Make sense? Just because we get satiated doesn't mean we stop eating. That's a whole nother thing. CCK. CCK also goes, what about stress? Yeah, stress is, hypothalamus is linked to the limbic system by the fornix. And the fornix just feeds all that information right into the hypothalamus. And yeah, everybody's stress is different. Some people stress eat. Some people stress stop eating. Yep, exactly. So it gets a lot more complex when you're talking about the brain. Oh, good old leptin. Yeah, I don't get to talk about leptin. So CCK actually talks to fat, and fat is what secretes leptin. You guys, zero testing on leptin, unfortunately. Leptin is definitely one of the most important hormones in the body. It's a part of the whole diabetes syndrome that's going on out there. But fat produces a hormone. Who'd have thunk? It's called leptin. The more fat you have, technically, the more leptin you secrete. Technically, the more leptin you secrete, leptin talks to the hypothalamus and causes satiation. So technically, the more fat you have, the more satiated you should be. But we all know that that's not always true. Leptin is also a key player. Oh, goodness. God, I wish I got it. Yeah, anyways, well, I just won't test you guys on this. Fat, again, now we're talking about fat. Fat produces this hormone called leptin. Leptin is one of the key triggering hormones in adolescence, in puberty. The more fat you have, the more leptin you produce. The more leptin you produce, that goes up to portions of your circadian rhythm that actually is going to trigger puberty. So what are we seeing nowadays? I don't know if you guys have even thought about this, but puberty is occurring earlier and earlier and earlier in our young. On average. There's always been people that enter puberty young, but nowadays it's pretty common for puberty to occur pre-teens. It's pretty common for people to enter puberty at 9. So what's one of the driving factors of that puberty? Fat. And we as a population tend to have more fat. Not a bad thing per se, but definitely has some risk factors that go with it. Does that make sense? Again, I wish I got to talk more about leptin. I used to work with leptin knockout mice, and whew, there's a lot of crazy that goes along with leptin. And leptin does talk to CCK. So CCK, other receptor sites for CCK that you guys need to know about. Pancreas. More specifically, in the pancreas, I'm talking about the acinar cells. And it's these acinar cells that secrete pancreatic juices, the ACEs, right? So these guys are the ones that secrete peptidase, different types of peptidases, different types of amylases, different types of lipases, and then nucleases. So now you can see that CCK plays a pretty critical role in the digestion of your food, because fats and proteins into the duodenum, now it's going to trigger the pancreas to secrete these juices into the duodenum. Make sense? And now these juices are going to help to digest that. Now, CCK also goes to the gallbladder. CCK is the hormone that triggers the smooth muscle of the gallbladder to contract. And when it contracts, it releases bile into the duodenum. And that's going to help with the emulsification of fats. Once the fats are emulsified, now these lipases here can digest them. If they're not emulsified, they can't be digested. I will talk more about emulsification today. I hope I got an hour left, so I got to get rolling. But we must have emulsification of fats for lipases to do their job properly. If you don't have the proper release of bile, if you've had your gallbladder removed, you really have to be careful about the amount of fat you eat. If you eat too much fat, your body simply cannot digest it properly. Because let's think about this. If you had your gallbladder removed, does that mean that the liver stops producing bile? Yes or no? No, of course. The liver is still producing bile. You still have your bile tracts. You still have your hepatic tract and your bile tract out, your common bile duct. You still have all that stuff. It's still entering bile down into the hepatopancreatic ampulla. You're just not going to be releasing as much in a bolus. You're going to be releasing a little bit all the time. Does that make sense? There's data that suggests that, Heather, but I don't have a complete answer for you, so I'm not going to make any definitive answers, okay? I don't know exactly the statistics on that. Yeah, there's definitely a lot of genetic and everything. Crazy enough. I mean, yeah, one person could live to 100 years old smoking cigarettes and eating bacon every day, right? Grumpy old men. And that's some true stuff. Awesome. Cool. Yeah, I was going to ask, so does anybody have their gallbladder removed? Andy, you're one. Does anybody else have their gallbladder removed here? Your mom? Yeah, I mean, you're fine. That's the great thing about gallbladder removal. You just have to watch your fat content of your food and eat much smaller amounts of fat in each one of your meals. If you eat too much fat, you simply can't digest it, and it will end up in your fecal matter, and that can potentially change the osmolarity of your large intestines and cause further downstream issues. But ultimately, it's not that big of a deal to have your gallbladder removed because the liver still do what it do. All right, let's move on. There are other hormones to learn about. Secretin. Secretin is a hormone released by the small intestines due to the presence of hydrochloric acid. Let's think about this the same way we thought about cholecystokinin. If acid is the stimulus, what's going to be the function of secretin? It's going to communicate to something else to try to reduce the amount of acid. Does that make sense? It's all about homeostasis. Homeostasis. So secretin, and we got some flow charts coming up for it. Secretin is stimulated by the presence of acid. Now it's going to communicate to the pancreas, and it's going to communicate to the stomach, and it's going to say, yo, pancreas, send me some bicarb. Because what neutralizes acid? Bicarb. Yo, pancreas, send me some bicarb. And bicarb is going to start getting pumped out of the pancreas into that small intestines. By the liter, almost literally. Lots and lots of bicarb is going to be shooting out of, we're going to call it shooting. It's not shooting, obviously, but being secreted by the pancreas. And then in the stomach, you're going to produce a little bit more bicarb on the stomach lining, but the primary role with secretin in the stomach is if you have too much acid entering your small intestines, let's just talk to that pyloric sphincter. And let's just slow down the amount of material that gets sent out to the duodenum. Does that make sense? Let's just slow it down. I don't need that much acid. There's too much. Slow down, stomach. Slow down. So it's going to do everything. It's going to activate somatostatin. It's going to start to inhibit acid secretion, and it's going to slow down the gastric emptying. Gastric emptying is the release of chyme through that pyloric sphincter. Motilin, I never test you guys on, but it's pretty easy. Motilin is all about motility. So basically, if you fasted for a little bit of time, that starts to actually stimulate your GI tract. How crazy is that? It requires fasting to move things through your system. You can't eat every 30 minutes or every hour and expect to poop more. It requires some period of time to get those intestines to do what they need to do to push all of that digested food through the system. GIP and GLP-1. These are the two hardest ones for students to learn, but I'm going to simplify it for you. I'm going to go ahead and test you guys on GIP and GLP-1, and their stimulus is carbs. Sure, there's other things, like GIP is also stimulated by fatty acids and amino acids, but I won't test you guys on that, okay? GIP and GLP-1, they have a commonality, and that is glucose. Sound good? Why this is important is because GIP and GLP-1, gastric inhibitory peptide, if we're getting too much food in the small intestines, what is our small intestines going to tell our stomach? Whoa, right? Stop. So it's going to inhibit that gastric emptying and acid secretion yet again. Does that make sense? Cool. Cool. It also communicates to the pancreas, and more specifically to the beta cells of the pancreas, because why, y'all? If I have glucose in my small intestines is quite aware that it's about ready to absorb that glucose into the blood. And what do we know? Exactly, Ruju. Good. What do we know is that an increase in blood glucose levels causes the secretion of insulin. This is a feed-forward mechanism. This is probably the first feed-forward mechanism that's really been pointed out to you in physiology. But this is a way for the intestines to prep the system. Your blood glucose levels haven't even gone up yet. You've just eaten a meal that's rich in carbs. And then your intestines start telling your pancreas to secrete insulin. Get ready, start secreting insulin, build up, ramp up. GLP-1 does the same thing. That's why I like to test on these two in a similar function. Okay, both of these are going to stimulate insulin release. Both of these are going to inhibit glucagon, and both of these are going to act back on the stomach to say, we got plenty, no worries, we got plenty. GLP-1 is unique that it goes up to the brain and also promotes satiety. So, cool. We got different slides for each one of these. Gastrin gets its own slide. Cholecystokinin gets its own slide. Secretin gets its own slide. So please look through those 3 slides. I just did this with you. For instance, with secretin we know is produced from the duodenum, and its stimulus is acid. The more hydrochloric acid that enters the duodenum, the more secretin is secreted into the bloodstream. And that secretin is going to go to the stomach and say, whoa, ho, ho, ho. So it's inhibiting gastric motility, gastric emptying, and gastric acid secretion. In addition, it goes to the pancreas and it goes to the liver. And it says, send me more bicarb. I need to neutralize this acid. Send me more bicarb. See or no? And when we look at that in a flowchart form, it becomes even easier in my mind, right? Acid in the duodenum causes the release of secretin. Secretin dumps into the bloodstream, and it goes places. One of the places it goes, for instance, is to the pancreas. And those duct cells of the pancreas secrete bicarb. Sodium bicarb now gets dumped into the pancreatic tract, the main duct. There we go. I don't know why I'm not saying the word. The pancreatic duct, the main pancreatic duct, and that goes to the hepatopancreatic ampulla and then out into the duodenum, which neutralizes that acid. Sweet. CCK, there's a flow chart for that on the right. This is not, this does not have everything, whereas this is everything that I want you guys to know about CCK is what we wrote down. So this particular flowchart does not have everything. However, it is the link to the pancreas. The pancreas has 2 major types of cells. This was part of yesterday's lecture, but I don't think I pointed it out. So let me go ahead and point it out right now. If you look at your lab objectives, part of what you guys need to be able to identify are acinar cells. And I don't think you need to identify duct cells, but they're the other major type of cell in the pancreas. Duct cells are pretty simple to think about. Okay, they are the ones that line the ducts. Sweet, they're called duct cells because they line ducts. Acinar cells are the #1 cell found in the pancreas. Acinar cells are these orange cells that are, surrounding this bulbous region. And if they're the #1 cell, what's the primary function of the pancreas? What's the primary function of the pancreas? Not to secrete insulin and glucagon. What's the primary function of the pancreas? Digestive enzymes, pancreatic juices, digestive juices, all of those words work just fine for me, okay? Yes, no, maybe so? What's the primary function? And what's the primary cell? They're the acinar cells. So it's the acinar cells that secrete those juices. Make sense? Nice logic that goes behind a lot of physiology if you just stop and think and stop stressing about whether you get the answer right or wrong. You're just like, oh, wow, shit, man, we got like 90% of the pancreas is made up of acinar cells, and the pancreas function is digestive juices. Therefore, the acinar cells must be doing this job, and that tends to hold true in the human body. Boom. Okay, cool. Let's see, what last one? GIP and GLP-1. These guys both are, all about glucose. All about glucose. They love the glucose, and they're feed forward because they start to stimulate insulin. Okay. 95, 45, 55. I don't know if anybody's watched that. Okay, so quickly, yet not quickly. My IQ 55. All right. Now, just to know what I'm talking about. So this flow chart, don't blow your brains up looking at this thing. Okay, everybody, just stop and look at me. Look at my eyes. Oh wait, never mind. It's online. Nonetheless, take a deep breath. This is not as crazy as it looks. So let me break this down for you. Break it down. Anyways, digestive joke. If you get food into your stomach, this top rectangle, I can talk, I swear, is the stomach. Where does food go to next? Stomach to the duodenum. So the next box down is the duodenum. Does that make sense? Okay, so that's the first thing to think about. The third box down is the pancreas, because it's always going to be communicating with the pancreas one way or the other. Now, what are these arrows? What are all these dang crazy, weird dashed arrows about? These are negative feedback loops. This is all about homeostasis. This is how complex, and this is just the beginning of the tip of the iceberg kind of thing of the homeostasis that your GI tract is constantly trying to do. It's crazy, huh? But again, you know 90% of what's here already. Actually, 100% technically. But we're going to tie in these negative feedback loops. So as food enters the stomach has hydrochloric acid. Hydrochloric acid has pepsinogen that has been activated. So we have pepsin in that stomach, and we have activated lipase from the salivary amylase. Yes, no, maybe so? We've also started to digest carbohydrates already. So let's churn, mix, make it into chyme, and then that stuff is going to be leaving the stomach through the pyloric sphincter into the duodenum via gastric motility. Motility, movement of the stomach, and that's going to end up causing what's called gastric emptying. It's going to empty small amounts of chyme into the duodenum. Easy peasy lemon squeezy there. That chyme is going to be communicating with everything. Enteric nervous system, we get some feedback loops just via a local intrinsic nervous control. What else is going to be a part of our chyme? So that's what's next. What's going to be in that chyme? You eat a big old bunch of food. Well, we're going to have everything from carbs, fats, proteins. Your stomach has released acid. And we cannot forget about osmolarity. We can't forget about ions. Remember, ions in solution create an osmotic solution. True story? Whether it's sodium, chloride, potassium, calcium, bicarb, all of that contains osmolarity. Because where does water want to be? Where that party's at. Does that make sense? Does that make sense? So this is the composition of that chyme, these yellow circles. Sweet. What we have down here is how, well, it's taking everything we've been learning and it's now tying it together, right? Which is the function of physiology. It's comprehensive. Now, what we have down here is what we've been learning up above. For instance, what do we know was the stimulus for CCK? Fats and proteins. What was the stimulus for GIP and GLP-1 that I asked you guys to learn? Carbohydrates. What was the stimulus for secretin? The presence of acid. So far, so good, everybody? What is the function of CCK? CCK goes all over the place. The hypothalamus, the pancreas, man, it even goes to the gallbladder. In the pancreas secretes more digestive juices. Sweet. The other function of CCK ties into this dashed blue line. And you'll notice that GLP-1 and secretin both go into this dashed blue line. that goes to the right side of this table, flowchart. It's a negative feedback loop. You got it. What you may also notice is that GIP and then osmolarity actually induces, we don't know what. Good old science, right? Science is always evolving. But osmolarity is going to end up triggering something. But what we do know about osmolarity is that it only impacts gastric motility. So depending on how osmotic your food is that you're eating, say for instance you just eat a boatload of popcorn, right? Highly delicious with all that natural butter that you put on it at the movies, and no salt at all as you shake all that flavored salt and stuff on there. Highly osmotic. True story? Anybody ever been surprised as you're sitting there with your freaking 14-gallon tub of popcorn and you're eating it and you're so excited to eat it, and then you make it like an inch down into that tub of popcorn and you're like, damn, I'm full. Like, what's going on here? I can't finish this popcorn. I don't know, that's me for sure. One of the reasons why is because all of that osmolarity blocks gastric motility. So your stomach stops churning as much and it stops emptying that food out. And it starts to fill and feel satiated. Crazy, huh? GIP only acts on acid secretion. It does not impact gastric motility. So the more GIP that's released, the less acid secretion you will produce. But gastric motility will remain the same. So I guess what I'm trying to point out and make sure that you guys see is that these two loops on the left trigger one or the other, never both. That's the way that this flow chart is set up. Whereas these loops on the right all act on both acid secretion and gastric motility. Do you guys see that now in case you didn't see it before? One or the other on the left, both on the right. Yes, no, maybe so? Okay, cool. Whew. All right, we got 30 minutes to get through the rest of this. We got this. What we need to talk about next is digestion. You guys are like, but we've already talked about, we haven't finalized digestion yet. We've gotten food down into the duodenum. Great, go us. Woohoo! We've looked at some of these hormones that are triggered because of the presence of these foods in the duodenum. Great. Pretty cool. Well, we ain't done. The duodenum is now, I'm pretty sure I said the duodenum, I stuttered, is now the place where the rest of digestion occurs. And not only that, we haven't talked about absorption yet. We got to get this stuff from the lumen of the digestive system into our blood. And that ain't no guarantee. Because if I'm talking about absorption, what other structures must I be thinking about? Help me help you. Faster. All right. Good. We got to be talking about transporters. I hope that starts coming faster to everybody. Okay? If we're talking about absorption, if we're talking about secretion, we are talking about protein transporters on the cell membrane. Okay? This is important. If you don't write down the transporters on your tests, you will get a lot of points off. A lot. If you're not writing the hormones, if you're not writing down the digestive enzymes, and if you're not writing down the different transporters that are involved in things, you're going to be getting a lot of points off. I'm just telling you that straight up because of how much I've emphasized it. Physiology is not telling me digestion will increase. It's telling me how digestion increases. It's telling me how absorption occurs. That's physiology. And that's the hard stuff, right? So please, please, if you are giving descriptions on a short essay or critical thinking question, be thinking this gigantic bold letter word, how. How does this happen? Don't tell me why it happens. Why did Brad Pitt and Angelina Jolie break up is completely different than how they broke up. Those are two completely different words. So when you're describing physiology, make sure you're describing how. And that involves everything we're about ready to go into. Proteins are big, huge chains of amino acids. Now we have to absorb amino acids. We don't have the ability to absorb peptides, polypeptides, and most importantly, most definitely not proteins. So what does our body need to do is break everything down from the protein level to the amino acid level. Does that make sense? How? We'll get there. Carbohydrates. Carbohydrates are called polysaccharides. Okay, poly meaning many sugars, polysaccharides. And we must make it all the way down to the monosaccharide level before we can absorb. Triglycerides, 3 fatty acids and a glycerol. We need to break these things down into smaller components. Monoglycerides, 1 fatty acid and a glycerol, and 2 free fatty acids. These guys right here are what run your Krebs cycle, by the way. So let's figure out how we're going to do that. And this table summarizes everything. So if you guys, by the end of today's lecture, are just like completely just flabbergasted, when you go to study, pull up this table, slow yourself down. I love this table because it works via a flow chart. And it works one macro or biomolecule at a time. So I like to start with carbohydrates or proteins, whatever. Right? And it's going to say, what's happening to carbohydrate digestion? Well, we start digesting carbohydrates where, y'all? Where do we know we're starting to digest carbs? In the oral cavity because of salivary amylase. So that's going to start to digest starches. Sweet. What about in the stomach? What are we doing to carbohydrate digestion with all that hydrochloric acid? Nothing. Hydrochloric acid has no impact on digestion. Let's get down into that small intestines. And the small intestines is going to be, at this point, because food has entered the system, so good old CCK is going to trigger the release of pancreatic juices. And in those pancreatic juices, there are pancreatic amylases. True story? Those pancreatic amylases are going to continue the digestion of those polysaccharides. We'll talk more about that, but that's how this table is set up. Once the polysaccharides are small enough, once they're down to a disaccharide level, we're going to need to break them from a di to a mono. And that's what the brush border is for. So your small intestines has a border of all these plicae circularis. You guys remember that from lab yesterday? The circular folds. And if you look at these circular folds, they're covered in fingers called villi, right? Plural is villi, villus is singular. And the villus is covered in simple columnar epithelial tissue that have microvilli. So yeah, circular folds, villi, microvilli, that is an enormous surface area. These microvilli on the surface of the simple columnar epithelial cell, this is called the brush border. And the brush border is constantly secreting different enzymes. I shouldn't say constantly, definitely triggered by portions of the enteric nervous system. So when I'm talking about brush border enzymes, it's this row right here, okay? So when you guys are taking the test, you'll have this table. It summarizes everything for you. Use it. As long as you can, right? You got a class that allows open notes. Use your open notes. So proteins, no protein digestion in the mouth. Boom. We're going to start talking more about protein digestion. Lipids, we get lingual lipase, but it's not activated until it hits the stomach. Now we're starting to digest some triglycerides. There are some gastric lipases that are also released, and then we're going to talk about pancreatic lipases in just a second. Okay. Ready, set, go. We're going to start with protein digestion. Protein digestion, and we're going to go a step back because I like to go a step back and remind you guys of things from time to time, but we got to go kind of fast here. We're going to say we have protein in the duodenum. Okay, this protein has already been partially digested because it's in the duodenum, so it was already partially digested in the stomach. The presence of partially digested protein in the duodenum is going to cause the secretion of what hormone? Practice makes perfect, y'all. What hormone is going to be secreted due to the presence of partially digested proteins and fats in the duodenum? Good, great. CCK, work on that one. I will test you guys on this one, so work on that fast trigger. Partially digested proteins in the duodenum, boom, CCK. CCK is now going to do a number of things, but in terms of its action on protein digestion, what we need to be thinking about is that CCK talks to the acinar cells in the pancreas. And then these acinar cells secrete pancreatic juices. And one of the portions of pancreatic juice, right, there are pancreatic amylases, there are pancreatic lipases, pancreatic nucleases, but one of the enzymes secreted from the pancreas are pancreatic peptidases. And these peptidases are going to help to digest this protein. Make sense? All right. So what peptidases are being secreted here? 1, trypsinogen. 2, chymotrypsinogen. 3, procarboxypeptidase. What do you guys notice about these peptidases? They are all inactive. Ogen, inactive. Ogen, inactive. And pro at the beginning of an enzyme protein name tells you that that is also inactive. So pro is just a different protein inhibitor, and that is inactive. So trypsinogen, chymotrypsinogen, and procarboxypeptidase, these are all inactive. They get secreted into the duodenum. All 3 of these go into that duodenum. Now, what's happening inside of that duodenum is we got the brush border. Okay? They get secreted into that duodenum, and the brush border recognizes the presence of these peptidases, and it's going to secrete an enzyme activator. There's a bunch of different names for this enzyme activator, but the number one, I mean, basically you guys can just call it enteropeptidase. Entero, meaning enteric nervous system. Peptidase, telling you that it is an enzyme for peptides. Enteropeptidase. So that's being secreted by that brush border. We'll come back to that in just a second. So what does that enteropeptidase do? That enteropeptidase activates trypsin. That's all this one does. Enteropeptidases are going to go back to this trypsinogen, and it's going to cleave the ogen off of the trypsinogen, making it so that all we have left is trypsin. Does that make sense? So now we have trypsin. Trypsin is basically the rest of the show. So trypsin is the guy now that is going to go and cleave the ogen off of chymotrypsinogen, and trypsin goes and cleaves the pro off of pro-carboxypeptidase, and trypsin, all three of these now that they're active, trypsin, chymotrypsin, and carboxypeptidase, these three things together are digesting proteins. So once we get trypsin activates everything else. Peptidase. And then all of those together are digesting proteins. Now here's the problem. You guys are like, what? We're not done yet? No, we're not done yet. We're just digesting these proteins down into like tripeptides. And peptide chains that are short, but they're not single amino acids yet. So what else are we going to need? More enzymes. Now, I'm going to stop though. I won't test you guys on the rest of these enzymes. Fair enough? Because I feel like you already have enough enzyme craziness going on here. And this is good. This is a great start to anybody's knowledge base on how we digest proteins. The rest of the proteins are labeled here. There are these carboxypeptidase, trypsin, chymotrypsin. You've already seen those. But there's also aminopeptidases. And these other dipeptidases and aminopeptidases, they're being secreted by the brush border. So once we get them down to these peptide chains via trypsin, chymotrypsin, and carboxypeptidase, now we need to take these 4s and 3s and 2s, and we need to cut them down to single amino acids, and it's the brush border that does that job. And if you know anything about amino acids, you'll know that one group is an amino end and another group is a carboxy end, and we have peptidases that will recognize specifically this nitrogen-carbon link in the carboxyl group, and we got ones that will specifically recognize the link between the nitrogen and carbon on the amino group. It's crazy. It's cool. If you're interested in it, it's all here. But you guys don't need to know it down to that level. So let's look at this picture real quick, and I'll do this in 1 minute. Ready? Pay attention. You ate food. Food is digested partially in the stomach. That is going to have proteins in it. Those proteins enter the duodenum. The duodenum now secretes CCK. CCK goes to the pancreas and off into these acinar cells, and the acinar cells are going to secrete trypsinogen, chymotrypsinogen, pro-carboxypeptidase. As they are going through the duodenum, they will encounter enteropeptidase. Enteropeptidase is going to activate the trypsin. Once trypsin is active, it's going to activate chymotrypsin and carboxypeptidase, and those three things together are going to take these oligopeptides, these partially digested proteins, and it's going to break them down into smaller and smaller peptide chains. The brush border finishes the job and gets them down to the single amino acid level. Once they're at the single amino acid level, we can absorb them. What do we know about absorption? Absorption of particles, molecules requires transporters. We got to have transporters. So what transporters are around for the absorption of proteins? The one that I will test you guys on the most is this guy right here. This is a sodium amino acid symporter. Remember what symport means. It's moving the two things in the same direction. Single amino acids are going to be moved from the digestive system tracts, from the lumen, into the cell. And it requires secondary active transport, kind of secondary active transport. It is a secondary active transporter, but the sodium concentration is mostly high, not through sodium potassium ATP pumps, but because of the food you eat. And that's what I'm trying to get at. But it's still a secondary active transport because amino acids are going to be going from low to high, whereas sodium is going to be going from high to low. Do you guys remember learning a transporter very similar to this earlier this semester? I hope the answer is yes, because we're about ready to get to it again. It was SGLT. Remember this one when we were talking about insulin and stuff? We got sodium glucose transporter. The SGLT took sodium and glucose, and it used sodium concentrations going from high to low to be able to move glucose from an area of low to high. Make sense? Well, here what we're dealing with is an amino acid transporter. It's a sodium amino acid transporter. And it's going to be moving sodium from high to low, and it's going to be able to move amino acids from low to high. So they're almost identical. This is the primary transporter that I will test you guys on, would be this one right here. There are other ones because we need to get small peptides like dipeptides, sometimes tripeptides, very seldomly tripeptides, but dipeptides can move in low numbers via a hydrogen transporter. But I tend not to test on this one. It's cool, it's important, but not as important as this one right here. The last thing that I want to, I guess, and then if we're talking about the basal surface, these are just passive carriers. So we have lots and lots of different amino acid carriers on the basal surface for pretty much each amino acid type. And it's just going to be facilitated diffusion. So as the amino acids build up inside the cell, it's going to go from high to low. Make sense? So the basal surface is really easy. It's just specific amino acid carriers. Versus this sodium amino acid transporter, symporter, it doesn't care what amino acid binds to it. It's going to be moving all amino acids with the presence of sodium. So a really cool way to think about this is, who here has ever eaten a protein that did not have salt? We all have at some point or the other, or it's not salted enough. What does it taste delicious? Or is it just a little bit meh? Everybody with me on this idea? That food tastes better with salt? Yes, no, maybe so? Why does our body do this? I love physiology because our body isn't stupid. It tells you salty food tastes better because salt is required for the absorption of that food. How cool is that? It's a real simple little idea. It's amazing. I agree. By the way, since I'm kind of running out of time, what about carbs? Whoever's made chocolate chip cookies and forgot the salt, might as well just throw those bastards away. Who's ever made a, or had one of those torts with the caramel drizzle and the big salt chunks on the top? It's just amazing, divine. So what about carbs? All carbohydrates that you eat taste better if there's salt there. You may or may not even realize that because a lot of your chocolates just have salt already in it. But all your baked goods add salt, everything, cakes, everything. Why? Because even those carbs taste better when you eat them with salt. So what are you starting to think about the transporter? What are you starting to think about the transporter? Help me help you. Don't waste too much time here. SGLT, guys. Right? It's going to need salt. Andy, you got it. Great job. Everybody else understand that? Our body is smart. It tells us food tastes better if we put in all the appropriate pieces of the puzzle. So let's go ahead and talk about these carbs. Carbohydrates are not an easy thing to talk about because, one, they're really polarizing. The world seems to be fighting a lot about carbohydrates nowadays, and for good reasons. We're going to bring one kind of polarizing piece of the conversation in as fast as I can. But the very first thing I want you guys to realize is that, carbohydrates typically come in a polysaccharide form. Polysaccharide is a big mini sugars. And the primary polysaccharide that we seem to eat nowadays is starch. But I got to remind you guys that glycogen is also just big long chains of glucose. And glycogen is received through eating meat, especially liver, because the liver stores a large majority of glycogen, but skeletal muscle also stores glycogen. So if you're eating meat, you are getting sugar. Stop thinking you're not. Your body can take these starches and these glycogens and it can start to break them down. Where do we start breaking starches down? Boom, right in the mouth with salivary amylases. Where do we finish breaking starches down? In the duodenum via pancreatic amylases. Make sense? Now, there are other sources of carbs. There's lactose, which is a disaccharide, so two sugars, and there's sucrose. This is good old, baking sugar. What most everybody loves, this sucrose right here. These are disaccharides. And maltose, you're not going to eat maltose. You're going to eat starches and glycogen, and those are broken down into maltose. So just a quick reference guide. Actually, it's all here. I'm not going to write it down. A quick reference guide to lactose, maltose, and sucrose. You can tell these all end in OSE, OSE, OSE. So these are disaccharides here, and they're sugars, hence they end in OSE. Lactose is made up of two monosaccharides, hence it's a disaccharide. And what specific monosaccharides make up lactose, galactose, and glucose? I'm not going to go back into a review of sugars. This hopefully was something that you had learned before coming to this class, but if not, again, you can study a little bit on it or you can reach out to me. That's not a problem. But if you look up these monosaccharides, galactose and glucose look identical. They have one side chain that's different. Only one side chain. So they're technically two different chemicals, but your body can utilize them the exact same way. Does that make sense? They are, as a chemical goes, as close as you can be to being the same thing with just one small change and a flip of an arm. That's it. Galactose and glucose. This is milk. Okay, I pointed at lactase and said this is milk. No, lactose is milk. So this is the sugar found in milk. Maltose is just a glucose and a glucose. So that should make sense because starch is just big, huge, long chains of glucose bound to glucose bound to glucose bound to glucose. So maltose is just a glucose bound to a glucose. This is awesome. Our body loves these. Glucose is what it really wants to use. It doesn't really care for lactose as much, and it can use it as energy. Fructose doesn't care about as much, definitely can use it as energy. It loves glucose, and we could use these over the long term. So the last one is sucrose. Sucrose is what we seem to eat the most. Everything that's processed is coming in as sucrose form. Sucrose is a glucose bound to a fructose. So our body can get these monosaccharides out. Fructose is just a pentamer. It's actually a completely different form of a sugar than glucose because of the way it folds in water. This is going to be interesting in just a second. But our body has to take it from the disaccharide level and bring it down to the monosaccharide level. And the way that it does is again with the brush border. So once we get these things broken down to disaccharides, and that's what amylases are for. Let's get these polysaccharides down to the disaccharide level. Now, as these disaccharides are passing the brush border has secreted other enzymes. The good news about these enzymes is they follow the names of the disaccharides. So lactase is required for lactose to be broken down into galactose and glucose. Maltase is required for maltose to be broken down into glucose and glucose. Sucrase is required for sucrose to be broken down into glucose and fructose. Anybody in here lactose intolerant? Moi, me. Yeah, so what's going on? You guys genetically aren't producing enough lactase. That's it. And if you're not producing enough lactase, the lactose isn't getting broken down into galactose and glucose. And therefore, can we absorb lactose? Do we have transporters for disaccharides? Yes or no? Everybody, anybody? We don't have transporters for disaccharides. I've mentioned that a number of times. We got to get it down to the smallest available unit. True story? So what's going to happen to that lactose? Well, if it's not broken down and absorbed in the small intestines, it's going to go through the rest of the system. It goes to the jejunum, it goes to the ileum, it makes it to the large intestines. What's living in your large intestines, y'all? And bacteria loves sugar. It's a fermentation process. Bacteria goes crazy over sugar, and it doesn't see a lot of sugar. You don't send it disaccharides normally because your body wants to absorb 100% of the sugars that you eat. But if you start sending it lactose, it goes through a fermentation process because bacteria is anaerobic. Fermentation, y'all. That means making beer. And a byproduct of fermentation to make lots and lots of ATP is carbon dioxide. And it starts to build up that gas in the large intestines. And that's why people that are lactose intolerant get so intense pain, so much intense pain, and then get all, gassy. Just don't stay in the same room as y'all if you've had some ice cream, right? Does that make sense to everybody? It's some pretty simple, fun little, relevant to life kind of thing. Trying to teach you guys that. The last thing I'll talk about, and then the rest will be up to you. Not too hard with fats, I don't think, personally. It just has to do with nonpolar versus polar. But the last thing that I will test you on, therefore I want to make sure to teach you on, is monosaccharide transportation. Monosaccharide transportation. What did I say about glucose and galactose? They are very similar to one another. What did I say about fructose? Fructose is a pentamer. It looks completely different than glucose and galactose. The structure is different. Yes, no, maybe so? What ends up happening here is because the structure is different, we need something different to transport it. So if I draw my cell, and I have the lumen of the GI here, and I have my blood over here, I need to get these sugars into the cell first and foremost. So if I want to take glucose and move it into the cell, or if I want to take galactose and get it into the cell, I need salt for that function. So what else am I going to be taking into the cell at the same time? Salt. Salt's going to be going from high to low, and glucose and galactose is going to be going from low to high. So what transporter am I talking about here? No, this isn't an amino acid, Ash. This is a glucose. Good try, though. I see why you did that. But I'm after that SGLT. Make sense? We're just time kind of thing. I know everything's going fast. So this SGLT is really important for the transport of glucose and galactose, but it cannot transport fructose. Fructose needs a different transporter. For fructose, it relies solely on a concentration gradient. Glucose requires sodium concentration gradients, so that's a limiting factor right there. Fructose, on the other hand, it's going from high to low. So fructose simply goes from high to low. Therefore, it's going to be going through one of those GLUTs. You guys remember the GLUT2, GLUT4? GLUT4 was insulin dependent. GLUT2 is found pretty much all over the body. We're going to be talking about GLUT2 in just a second. Fructose can go through either GLUT2 or GLUT5. But the deal with GLUT2 is the honey badger, love honey badgers, of our glucose transporters. It don't care. It'll take all monosaccharides through it. So if we had a GLUT2 on the apical surface, then glucose, galactose, and fructose would all be able to go through it from high to low. But glucose concentrations tend to be low, so we can't use GLUT2 on that apical surface. But we still need to get fructose through, so instead we use GLUT5. GLUT5 is a glucose transporter that only transports fructose. That's it. That's all. And since it's GLUT, it's going from high to low. It's passive diffusion. Well, facilitated diffusion. This is GLUT5. In every case so far, though, what's going to happen to the concentration of glucose and galactose and fructose inside of these cells is it's going to start to increase, and eventually we just want to get this stuff into the blood, and we're going to go through a GLUT2. So on the basal surface, there's a GLUT2. On the apical surface, there's an SGLT and a GLUT5. Dr. H, why is this so important? Why are you making us learn all this? Who's ever heard of high fructose corn syrup? If you haven't, your head's probably been underneath a rock a little bit. You know, it's kind of a big deal. This whole high fructose corn syrup, processed foods. There's a big argument: feed your kids high fructose corn syrup, or don't feed your kids high fructose corn syrup. What's going on there? Well, high fructose corn syrup is 60% fructose and 40% glucose. That's what's going on there. So basically, it's a sugar, right? Like sucrose. Sucrose is 50% fructose and 50% glucose, but now we're taking high fructose corn syrup, which is 60-40. So there's more fructose. Why is that important? The companies will tell you a sugar is a sugar is a sugar. Is that true? Yes. All monosaccharides, no, that's actually a true story. They're not lying to you. A sugar is a sugar is a sugar, right? All these monosaccharides are used in the body the same way. They're all used to make ATP. So a sugar is a sugar is a sugar. What they're not telling you is that your body utilizes different transporters to get the job done. So now if I have a GLUT5 that relies on a concentration gradient, and you're taking in high fructose corn syrup, what just happened to the concentration gradient for fructose to get into the cell? Increase, decrease, no change. Does that make sense, y'all? The concentration gradient just went up. So what's happening to the amount of fructose that's entering the cell and the rate that it's entering the cell? Increase, decrease, no change. This one over here relies on sodium. It's slow. It's going to have to bind sodium. It's going to have to bind glucose or galactose. It does its thing. It's going to dump glucose into the cell. It's rate limited. Fructose ain't rate limited. It's just rushing into the cell completely based on a fructose concentration gradient, and then moves right through the GLUT2 based on a concentration gradient as well. So what's going to happen to the rate and the concentration of fructose in your bloodstream? Increase, decrease, no change. What's going to happen to the speed that insulin is triggered for release? Increase, decrease, no change. What's going to happen to the amount of energy that's going to get stored away because there's more energy available now at a faster rate? Increase, decrease, no change. I mean, so this is, I'm not going to go get into all the politics of it, but the physiology behind it is pretty simple. High fructose corn syrup definitely causes an increase in the amount of fat deposition, insulin secretion. So is it linked with diabetes type 2? Yeah, you better believe it is, okay? Tons of science points to that case. Why? Because of the rate sugars are getting into the bloodstream. Does that mean that I don't eat high fructose corn syrup? Of course not. I still do. I'm just human, and stuff tastes delicious. Do I still give my kids high fructose corn syrup? Yeah. But do I think about it, and do I try to limit it? Sure, I do. So that's just my take on it all. All right. Done. You guys just work on the same idea, but with triglyceride digestion and the absorption of fats. Don't forget, if I get a fat droplet into this duodenum, CCK is going to be secreted. CCK, pancreatic juices and lipases, gallbladder is going to secrete bile. So make sure you guys are looking at how this emulsification process works, and let me know if you're having trouble, because I can help. Wait until the next unit, Tariq. The physiology is so comprehensive. It's all about building the story. It's all about going big, right? Yeah, you guys are learning that. You know that. You know that. Let's see here. Test I will release tomorrow. You guys know everything's due tomorrow, extra credit. Tomorrow at midnight. Saturday, homework is due. Sunday, lab practical and lecture exam. Text me if you have any questions, all right? Thank you, everybody. You guys are working your butts off. I know this ain't easy. Keep answering questions via how. Okay? How does this work? Don't say just because I ate a meal that I am absorbing proteins. It don't work like that. You guys rock. Keep it up.